If you're basically a meat eater, you can be very glycine deficient. It's a matter of balance. When you eat too much methionine, your body treats it as toxic.
So what do you say to people who just say, oh, the body's making enough glycine?
You don't have to measure the glycine in your blood, you can tell by its effect on inflammation. The only time glycine can really kill you is if you have an inborn metabolic disorder where you can't get rid of glycine. It's gotten a lot worse because of dietary habits, because we eat the meat and we throw away the bones.
The other one that you always hear, that I always get asked about, is that glycine will feed cancer cells.
Even though meat eaters eat more glycine, they have less glycine in their blood than vegans do. Glycine is as fundamental as it gets.
Fundamental, foundational. This episode today is all about busting glycine myths and answering the most common questions we've got. And so we're here with Dr. Brind. He's a glycine expert, even though he hates when I say that. Yale-trained, NYU. He's a biochemist. He has training in immunology and nutrition. It's a very interesting confluence of training that he has. But we're just going to jump right in. The first myth that I want to bust, it comes directly from one of your comments, and it says, neither of these two individuals know anything about glycine metabolism. Even healthy, consuming an adequate, balanced diet makes copious amounts of glycine. There is no glycine deficiency epidemic. So, Dr. Brind, why don't you start there? Tell us why you know a thing or two about glycine metabolism, and why it's unfair to say there's no deficiency epidemic.
You gave my background. I mean, I am a biochemist. I have a degree in basic medical sciences with major in biochemistry, minor specialization in physiology and immunology. And the most important role of glycine, I think, that bears on our everyday life that we can sense is the related to most chronic disease and inflammation. That has to do with glycine's role as a cellular physiological agent in the immune system. Physiology, immunology, biochemistry, right? So that's how it's just funny how all of this came together. My areas of specialization in my education, which was concluded in 1981, my formal education, and I got my PhD in 1981. It was, all of this started coming together about glycine in 2007, 2008. So it was almost 30 years before all of this came together. But it certainly comes in handy to be able to draw these threads together, rather than what's the trouble with modern science and modern scientists is this sort of tunnel vision kind of specialization. And nobody, you can, it used to be when I was a young scientist, it was bad enough because you would have all of these, it would be as if you have all of these tree specialists working on this kind of tree and that kind of tree, and nobody realized they were working in a forest. Now it's, you have a leaf and bark and root specialists, and nobody even realized they're working on trees, let alone in the forest. So they're not really connected with what else is going on. So now, the mindset that produced that comment, I think there was another comment like it, which talked about, there's another one on the internet where he says, want to see a great source of glycine here? And he has a chicken leg. I just got so many, grams of glycine in that bite, and it's, yeah, that's one of the myths that I busted in my book. The Glycine Miracle was about eating enough of these amino acids and high quality and all of that kind of stuff. And that is that you can eat more glycine than anybody else if you're a carnivore and you're eating nothing but meat and fish and poultry all day long, muscle meats, which are loaded in methionine and they're relatively glycine poor. However, because they're so high in protein, you still get a lot of glycine. So they say, oh yeah, you need 10 grams a day, and you get a third of it or something like that just from having a couple of chicken legs or something like that. It's a matter of balance. So, for example, I was saying 10 years ago when talking about diet, that if you're basically a meat eater, you can be very glycine deficient because muscle meats are high in methionine and unarguably essential amino acid. But because it's essential, your body has so many ways to recycle it and regenerate it that you really need very little. And we all learned in biochemistry graduate school, we all learned about the methionine cycle. One of the great cycles that biochemists are trained on is the basics of it, which is how your liver regenerates methionine, the main way that it does that, called the methionine cycle. So everybody knows about the methionine cycle and how your liver regenerates methionine. What they don't know, which more recent research revealed, is that when you eat too much methionine, your body treats it as toxic. So after you've eaten that cheeseburger, for the next 2 hours, your liver is busy getting rid of as much of it as it can, pretty much. And then over the next couple of hours, then you recycle it, and the glycine cycle and the methionine cycle goes to work. But you have kind of the reverse process, and you're actually clearing methionine. Now, the relevance of that to glycine is that for every molecule of methionine that is cleared, that is gotten rid of, you have to use up 2 molecules of glycine. The key enzyme there is glycine N-methyltransferase, which takes a methyl group off of methionine and puts it onto glycine in order to get rid of it, leaving you with methylglycine or sarcosine, which then gets again recycled around. But the glycine is used up in that way. You end up, this is what I predicted 10 years ago, that you would end up, if you were basically a meat eater and you eat meats the way most of us eat meats, that is, you cut the muscle meat off the bone, you cook and eat the muscle meat, and you throw the bones in the trash instead of the soup, like everybody's grandma did. I don't care what cultural background you come from, your grandma threw the bones in the soup, not the trash. You didn't waste it, and that's where all the glycine is. So that imbalance means that even though meat eaters eat more glycine, they have less glycine in their blood even than vegans do. Well, lo and behold, it was about, so I must have been saying that for about 12 or 15 years, I think. And then lo and behold, to my surprise and delight, 10 years ago, a group at Oxford did a study, part of the so-called EPIC study, European Prospective Investigation into Cancer, I think, which is, a multinational European study with lots and lots of money, billions of euros, millions and millions of euros, and, thousands of participants in different phases of the study. So this group at Oxford was interested in amino acid metabolism. So they took 100 vegans, 100 lacto-vegetarians, 100 fish eaters, 100 meat eaters, omnivores. Each one of them, they cataloged everything they ate, so they could calculate exactly how much of each amino acid they consumed every day. And then they took a measurement of their blood plasma to look at the free amino acid content of the blood plasma to see if, is there a correspondence? And they found out that there was, yes. If you eat more histidine, you have more histidine in your blood. If you eat more lysine, there's more lysine in your blood. If you eat more methionine, if you eat more, let's say, phenylalanine, there's more phenylalanine in your blood, except the one real outlier that they couldn't explain, and that's key, that they couldn't explain it, was glycine. They found that the meat eaters ate the most glycine, and yet they had the least in their blood. And we're talking about 15 to 20% differences, not 2 or 3%. These are substantial differences. So if you eat a lot of meat and you don't make up for it by consuming either a glycine supplement or having, eating, the bone part through a bone broth or collagen or gelatin product, that can offset that, you end up depleting your body of glycine. They showed that that's what happened, but yet they couldn't explain it, and that's because all of these people who supposedly know about, glycine metabolism and so on and so forth, they don't, they take a very simplistic look, and they don't really tie together from glycine metabolism how it's related to methionine metabolism, and then it's not just a matter of, that's simplistic, which is to say, oh, how much glycine you have in your blood? Well, it's how much you eat. Well, that was their simplistic hypothesis, that everything would be simplistically, you eat this much of amino acid A, you have this much amino acid A in your plasma, amino acid B, C, D, and so on. It didn't work for glycine.
Yeah.
And that's why, because of this reciprocal arrangement. How do I know? Because I predicted it. And
you studied it for decades.
And I studied it, yes. This biochemistry is known.
Yeah.
A big irony that I've never mentioned on your podcast before was that I did my first publication with glycine was an abstract that I presented at the Federation proceedings, the Federation of American Societies of Experimental Biology, which has been going on for quite a long time. So my paper was using glycine supplementation as a way to mimic methionine restriction. It turned
Yeah.
out not to work the way we thought it did. That was a hypothesis we certainly had to modify. But what was interesting is that when I went back and researched the reciprocal relationship between methionine and glycine. Now remember, here I posted this abstract, published the abstract at the Federation meetings in like April or May, the spring of 2011. And I found that there was an abstract showing the reciprocal relationship in the diet, just an abstract, published where at the Federation meetings at the same national conference in 1951, exactly 60 years to the day before
Wow.
I published this finding in rats, another group had actually published a similar finding about how in doing growth studies, because to figure out, livestock feeds and all that kind of stuff, a lot of research goes into that. They noticed this reciprocal relationship that it said that glycine can alleviate methionine toxicity. Because if you give too much methionine, the way it works with these nutrients, you give them more and more and more of this amino acid in the feed and they grow better, and then after a certain point, they grow worse. So that's the toxicity. Too much methionine was toxic. It was actually slowing down their growth. So my paper basically was essentially following up on exactly that biochemistry 60 years later to the day. I just thought that was... The authors' names were Solomon and Berg. I remember very well coming across that abstract. At the same meeting, 60 years later, here I'm basically showing the same thing, because there's nothing new under the sun. That's why I don't like to, be called an expert on this and that, because, God made it all. When you discover something, everybody acts like you invented it,
Yeah, and I love that. I love that humility. And you're saying this is a long history. You've been a good part of this history for a long time. So take us to like 2009 too, and maybe before that, when we realize even without considering how much meat is in your diet, we're still operating at a deficiency. And so what do you say to people who just say, oh, the body's making enough glycine? We don't
How do you know it's making enough? Because everybody says so. Because when you go on Quest Diagnostics website and you look at their normal range, or any other diagnostic company, I'm not singling them out. They're all very good in terms of defining what a normal range is. But again, you have to not be simplistic about it and just look a little deeper and say, what does it mean that the normal range of glycine is between, let's say, 150 and 350 micromolar? It means that's what most people have. Does that mean it's the optimal healthy one? Who knows? It's just what most people have. Well, it turns out it's not so healthy. You really need over 500. Somewhere in there, over 500. It's kind of it's not a hard and fast number. I know mine, when I took 40 grams a day, it leveled off at like 730 or something like that.
40
So
grams.
mine is probably somewhere between 500 and 700, and I know it's enough. And you don't have to measure the glycine in your blood. You can tell by its effect on inflammation. And the reason is that's because its effect on inflammation is as a physiological gatekeeper for the cells that actually cause inflammation. It's got to be the solution that these cells are bathed in the lymph in the body, or the plasma in the blood that the cells are bathed in. Glycine needs to be present in a very high concentration, and it's a lot more than you need just to make proteins, because glycine is used as a building block to make proteins. So you can have a lot less glycine than is really necessary for this key immune function of regulating inflammation, and not have any deficit in your body's ability to make proteins, even to make collagen. So that's why, your body can make it. Nobody has protein manufacturing deficiencies, in the normal population with a normal level of glycine. So non-essential, the normal range is natural and it's fine and everything like that. But glycine has many roles. And the key role in terms of inflammation is a physiological one, which requires a lot more glycine than the glycine you need just to make protein. And so that's why a deficiency is easier to get, and why it can go unnoticed for so long, because it's subtle. Chronic inflammation is something that you don't feel it in your arteries, and then all of a sudden 40 years later you get a heart attack. How'd that happen? Well, what was going on was unbeknownst to you. It was not palpable, whereas inflammation in your joints is very palpable, because that's where pain happens and swelling happens and all of that if things get inflamed. A key myth about glycine is that for someone who talks about the biochemistry, its key role is not really a biochemical role. So that's one myth.
Yep, that's an amazing myth to bust.
Glycine's effect on inflammation is a biochemical effect. It's not. It's a physiological effect.
Yeah, that makes sense. And so the another myth is when you think about glycine, that its primary benefit is for sleep.
You know, glycine's so important, and you always have a lot in your body, even if you don't eat any. Your body makes some, and if your body didn't make any and you didn't eat any, you'd drop dead very quickly. So, what do you say is the most important? You know, I mean, the inflammation thing is the most important in terms of chronic disease, because that's the subtle thing that builds up over time. But in terms of, you know, day to day, certainly, if you can't get a good night's sleep, that's pretty fundamental. And if you take glycine and now you can get a good night's sleep, that's pretty important to, you know, correct that imbalance and get your glycine levels right. So the way that works is, that's more recent research, I would say, in the last 10 years, and it's not even completely understood. But in terms of inflammation, there's that glycine receptor that I talked about, which is the same receptor that you find in the central nervous system, where glycine acts as an inhibitory neurotransmitter, kind of slows things down, acts through this so-called classical glycine receptor. Well, there's another receptor called the NMDA receptor, N-methyl-D-aspartate. It's a receptor that requires glycine as a co-activator. So it's a co-ligand that it's needed also to activate this positive receptor, this excitatory receptor, the so-called GABAergic receptor in the central nervous system. And that is necessary to be activated in order to promote what they call non-REM sleep, you know, deep sleep. And it's a very interesting experimental technique that they do this in like rats and mice, where they actually inject, you know, into the brain, to different parts of the brain, they'll inject glycine. Well, what happens if we inject it here? What happens if we inject it here? What happens if we inject it here? What happens? You know, different nuclei, you know, which are essentially collections of specific neurons, which are all associated. Some are associated with speech, some associated with emotions, some are associated with sleep, some different things. So there's a nucleus called the suprachiasmatic nucleus, and you have these neurons which are excited by the N-methyl-D-aspartate, NMDA receptors. And what glycine does there, the glycine's action there, is it stimulates the lowering of body temperature and the promotion of non-REM sleep, deep sleep. The way it does that is it causes peripheral vasodilatation. It causes the capillaries in your skin to dilate. What happens when that happens? They release heat. Your body temperature goes down. So it actually decreases your core temperature, which is important for deep sleep. And then it also causes a muscular dystonia or atonia, which means your muscles don't work. And so that also promotes this kind of deep sleep. By the way, I never mentioned this before also, but a hypothesis just occurred to me about that, and that is, and I don't know how much of a problem this is with somnambulation, you know, people who walk in their sleep, sleepwalkers.
Oh.
I suspect that glycine might cure sleepwalking.
Which is fascinating hypothesis that I will test, because I have two friends who do.
How do you have two friends who don't take glycine supplements yet?
Because they're family and they don't always listen to me.
Oh,
I do have that family. I have that family.
Right. Okay.
This is exciting. I love that theory. And I wanted to add too that I was reading research this morning on schizophrenia and OCD. There's a really interesting case report on OCD which shares some kind of NMDA receptor. There's theories that it's involved anyway, where a guy basically couldn't get out of his house, and he had like mirror issues and body dysmorphia, and they started supplementing. Well, first of all, he tried a lot of things, a lot of different antidepressants and a lot of different antibacterials and
antipsychotics, probably. Yeah.
Antipsychotics, yeah. But eventually got into, I think it was 50 and up to 60 grams of glycine. Now, within the first 30 days, he was out of his house. Eventually, he went on to go back to school and graduated, I believe, with like cognitive scores in the 90%. So this was like a really dramatic recovery for this specific person. And also, they're looking at it, there has been surprisingly little research in anxiety. But anyway, this NMDR receptor thing, I'm really excited.
NMDA, yeah. That's also implicated in glycine toxicity, and the only time glycine can really kill you is if you have an inborn metabolic disorder where you can't get rid of glycine. And this is called NKH, non-ketotic hyperglycinemia, and that seems to work through the NMDA receptor. I have a hypothesis about that too, that there's another supplement called DHEA, which is an adrenal steroid that's not a hormone, but it can be made into hormones. And the NMDA receptor, I saw just one abstract about it, seems to be protected by DHEA. And this stuck in my mind because DHEA is an adrenal steroid which is very, very high in utero. And as soon as you're born, as soon as that placenta is disconnected, it drops like a rock. And then DHEA does not rise again until you're 8 years old, so-called adrenarche, right before puberty. And the reason why that stuck in my mind is because people with NKH, these are kids, they're born normal. Their intrauterine development is normal. They're born normal. And then they start to go downhill after a couple of months. They start getting seizures and so on. And there are techniques to try and get rid of glycine from the body and all of that. But it seems that DHEA, if that actually can protect the NMDA receptor from glycine toxicity, which is when too much glycine is there, that might be an answer. But I wrote some emails to the main doctor. His name was Dr. Van Hove, I think, who was the big kahuna in terms of, NKH. I suggested this to him, and he was like, oh, but it's so difficult to get clinical trials. And clinical trials, it's an over-the-counter supplement, and these kids die anyway. A horrible death when they're a couple of years old. I mean, what are you talking about? Give them DHEA and see what happens,
I love that idea.
Sometimes experimentation is just so much of a no-brainer, but these days, not only is medicine so hyper-specialized, but it's almost completely paralyzed by, medico-legal concerns. Who's going to sue me for this or that?
It's so true.
Good thing I'm not a doctor and I don't give medical advice, And I'm also selling a product that's really harmless. So
Yeah.
that's what I've been doing it for 15 years without any difficulty.
Yeah, so that's another great place to share is that glycine, like you said, for some people with this inborn genetic mechanism, it might be dangerous, but for the general population, it's very, very safe, even at very
Well,
high
it's not
doses.
even dangerous for them. It couldn't compare to how much their body is making anyway.
Okay,
It wouldn't make any difference if they ate glycine or not.
got
People
it.
with NKH, I'm sure. It's very safe. Now, it's funny, I'm always looking at glycine materials, and I just came across for the first time an actual estimate of the LD50, how toxic is glycine.
Hey,
And
yeah.
it's just about 5 grams per kilogram per day,
Okay,
or in a
okay.
single dose. So you know what 5 grams per kilogram is? So I weigh 75 kilograms.
Yeah.
Right? So we're talking about 375 grams.
Wow.
That's
Yeah.
about 12 ounces
Yeah,
of
for me it'd be like
high glycine
250.
powder. Chug-a-lug. You can't eat that much. You can't
Right.
eat enough to do any real harm. And as I think I mentioned before, that the only literature that shows how high levels can really get is from the urologic surgery literature, because of this technique, this surgical procedure called a TURP, transurethral radical prostatectomy, which is used for benign prostatic hypertrophy. It's like a roto-rooter through the urethra. It's kind of a very bloody procedure, kind of a very crude procedure. In fact, it was perfected over 100 years ago, and they still do it because it works, and because men tolerate it surprisingly well. And that the reason is because they're basically pouring, as an irrigation solution, 2% glycine right into their veins, right into an open wound. So sometimes the concentration in the bloodstream can get to like 100 times normal. And I think in a few cases it caused blindness that lasted for a few hours, and then it all resolved, and no one ever got anxious about it. That was the key thing in these papers, is that even though some of them had this side effect of blindness, it's like they weren't troubled, they weren't worried, they weren't paranoid. When you talk about paranoia, schizophrenia, if you have enough glycine around, maybe that's something that a real pharmacological amount in the nervous system can do that. And I know the where they tried that clinically in these trials. They gave people 60 grams a day of glycine. And how did they administer it? They administered it by taking it like a teaspoon's worth, or which is like 5, at a time, like 4 times a day, whatever it was. And they said they put the powder under the tongue. And I thought to myself,
Yeah.
what kind of crazy way is that to administer something that naturally has a sweet taste? Can't you formulate something that tastes good? So I did. It wasn't difficult. So I made sweetamine with a little tweaking to make it taste more like sugar. One of the things that's in there is I put a little bit of erythritol. We talked about that before. Erythritol is a natural sweetener. Unfortunately, the article that sort of raised some alarm about it called it an artificial sweetener, which it isn't. It's a natural sweetener. Your body makes some erythritol, very little. Well, I put very little actually in sweetamine. Not enough that should alarm anybody, because a paper came out from the Cleveland Clinic a few years ago saying, erythritol, it's not so safe as they say. The trouble with erythritol is that unlike many other sweeteners like stevia, like monk fruit, like aspartame, which are very, very, very sweet, like 100 times sweeter than sugar, erythritol is a little bit less sweet than sugar, similar to sugar. So they use it like sugar. So in a can of soda that's sweetened with erythritol, there's 30 grams of erythritol in it. 30 grams of anything is bound to be trouble unless it's really, 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 really safe. Well, the FDA said it was generally recognized as safe up to 80 grams a day. And for most people, it probably is. But what this research showed is that if you take 30 grams of it, you can still see a day later, you can still see that it makes platelets more likely to aggregate. So you think, well, then it may increase the clotability. And if it increases clotability, then maybe it can increase the risk of stroke and heart attack. So they went back through their records and said, yes, people with erythritol, with erythritol in their bloodstream were more likely to have strokes and heart attacks. But again, that's because erythritol is used in bulk. And in fairness to the manufacturers who actually use it in dietary products as a sugar substitute, still better for you than sugar. All right?
But what is it
30 grams of sugar in a can of soda. So, but the thing is, in sweetamine, in every packet we have 800 milligrams, 0.8 grams, so less than a quarter teaspoon of erythritol in each packet. So it's not enough to be concerned. And even if you thought it had enough to make your platelets more sticky, guess what makes your platelets less sticky? Glycine. And the irony here is that the same group that raised the alarm about erythritol also raised the found out that glycine actually lowers the risk of major adverse cardiac events. They published that in 2015. The group from the Cleveland Clinic. So glycine, it's not only good for you, but if you put in a little bit of erythritol, erythritol is not the sweetener in sweetamine. Sweetamine is naturally sweet. You need to tweak the sweetness to make it taste a little more like sugar and get rid of the unpleasant aftertaste that's natural in glycine.
Yeah.
So anyway, that's, but because people react to headlines, and in fairness, they're not PhDs in biochemistry or basic medical science. And so, people tend to react to headlines, and it's natural to want to avoid anything that's associated with harm.
Yeah.
So, and since I sell a commercial product, since sweetamine is a commercial product, you have to respond to market demand. So we'll probably end up taking out the little bit of erythritol that we have in there anyway, and nobody's going to notice the difference in taste.
And I love your openness to that. And also, like you're saying, if you're having, it's the context too. Erythritol with 8 grams of glycine, different.
Yes, and, well, a small amount of it,
yeah.
Right.
So interesting. But I love that you're kind of responding to the comments too. Like, I think that's great. And what is cool about sweetamine is that we hadn't even mentioned this, but 10 grams is what you recommend in order to get to that 500, the level of 500 in your blood.
Yeah, roughly. But don't forget, the 10 grams includes the 2 or 3 grams you get from a typical diet. So that's why I put in 8 grams.
8 grams.
Yeah,
just so that we know how many.
Yeah, there's also taurine in there. Taurine is another beneficial, very fundamental amino acid that has a sulfur in it. It's a sulfur amino acid, so it's a good source of sulfate, which, because of things like glyphosate, Dr. Seneff says we're all deficient in sulfur, which may be true. But taurine is also can work like glycine. It can interact with the glycine receptor, and it's particularly good for muscles and heart muscle, and it's just an all-around very good molecule that your body makes, but in could make more of it. It's not like your body needs to take in sulfur-containing amino acids like methionine. Okay,
Yeah.
that's the only one that's really essential. But from methionine, you can make cysteine, you make taurine, you make all of those others. But adding a little taurine is very good. So so that's in the formula, but that doesn't have any effect on taste. That's pretty much what sweet amine is. What I'll probably do is add back some. I used to have proline in the mix. And proline, also a very good amino acid, also very high in collagen, and also has a kind of sweetness that tends to neutralize the aftertaste of glycine's sweetness. So I'll probably, a little sneak peek. But that's, it takes a long time, with supply chain lag times and whatnot. Look for it sometime next year to have an erythritol-free product, but it'll be just as good. And it will alleviate any fears that people have from the negative headlines that erythritol's unfortunately been getting. But those headlines don't really apply to glycine. There's really no harm at all.
I love that. Thank you for that nuance, and I'm really excited about that. And I just wanted to say, I'm just going to throw this in there, that we've done an episode about glycine and glyphosate with Dr. Seneff, in addition to the sulfate that she thinks. So
Right.
your supplement could theoretically also help potentially knock out glyphosate if that
Yes.
is in
To
fact
the, right, yes. To the extent that hypothesis applies,
right.
and as Dr. Seneff says, she's never got the money or knows of anyone who's tested it formally, but there is a lot of circumstantial evidence that one of the ways that glycine is toxic, we know that it's harmful, even though the FDA said that it wasn't. I don't know if they still do. We noticed that Monsanto is now taking glyphosate out of Roundup. They still use it as a drying agent. What they do is, these herbicides are supposed to kill weeds. But what some bright plant biochemist figured out, that you can save money in agriculture, save money in harvesting wheat, if after the wheat is mature, if you then apply a herbicide that actually kills the wheat, because then the plant shrivels up and dries up, and then it's much easier to harvest.
Yep.
Instead of waiting an extra couple of weeks for the plant that had the wheat to die off and so on. So they call that a drying agent, but it poisons it. The thing is, one of the ways, if not the principal way, that it poisons people is that glyphosate is also a simple small molecule that may be incorporated into proteins as amino acids are taken one at a time as specified in the chain of DNA and RNA to make a given protein. If it calls for glycine and you have a lot of glyphosate around, then one out of however many hundreds of times or tens of times or whatever it is, it will take a molecule of glyphosate and incorporate that into the protein chain, and then now you have a clinker protein that's either useless or almost useless or causing some other kind of harm. And so to that extent, the best way to get rid of it is to flush it out with an excess of glycine. So if you have, double or triple the amount of glycine that you normally had in your cells, when protein is manufactured, then presumably that will dilute the effect of the glyphosate and lower its toxicity.
Yeah.
That's the same theory that they have for like iodine. They take iodine pills for radiation poisoning in the case of nuclear war. It's, right? It's the same thing. You take in because your body, iodine is a relatively rare atom that your body actually needs. It's essential. It's an essential mineral. So that means that your body is wired to say, ooh, an atom of iodine. Your thyroid is like an iodine sponge. There's a process that works in the thyroid gland that I teach my students endocrinology, I call it iodine suction. It literally sucks iodine out of the gut, out of anywhere it can find it in the body. When thyroid hormone is metabolized and broken down, the iodine is, snatched back, it's clawed back from the circulation as much as possible. And in fact, when you have a goiter, when iodine deficiency causes a goiter in a large thyroid, the thyroid can actually get enlarged to like 200 times its normal size. That's why the neck can swell up like this, and the body's actually okay. It's just amazing what it can do in order to get every last atom of iodine that it can. So the thyroid gland is so good at concentrating iodine that it's great if there's a radioisotope of iodine that is toxic that you want to protect yourself from, you just have non-radioactive iodine, and that gets rid of it because it dilutes it out. Not to mention that nobody does surgery on the thyroid gland anymore. You have cancer of the thyroid or a tumor of the thyroid's not working. What do? They give you a cocktail of radioactive iodine. Why? That's toxic. Doesn't it destroy tissue? Yes. But it's so efficiently taken up and sent right to the thyroid gland that it just burns up the thyroid gland without damaging anything else.
Wow.
It's amazing. And most people, it's fine. They tolerate it and they get a little bit of radiation exposure from it, but not much compared to the actual thyroid gland, which is fried intentionally. That's the easiest way to get rid of it if you want to surgically remove it.
Wow, that's
Then you got
fascinating.
to take thyroid hormone instead of iodine as your essential nutrient. But
Wow.
it's the same thing. So we're getting back to glycine. It's the same kind of idea of diluting the toxic substance by taking more of the non-toxic one, and in fact the essential one, taking more glycine. So it may, theoretical now, it may lessen glyphosate.
Yeah, and there's a Dr. Klinghardt who is, I believe, a toxicologist, and I've tried to get him on the show, but he used 2 4-gram doses per day, and I believe he did before and afters in glyphosate testing, and I think he used other supplements as well, in addition to glycine. But it was funny because it was 4 grams twice a day, which is 8 grams, which is your supplement.
That's it.
Yeah,
Half a
so
packet of sweet amino in the morning and half a packet at night. Yeah, a lot of people
yes,
take it to divided doses that way. Yeah.
yeah.
So
So
did he publish that? Is that a?
I don't know that he published it. I listened to an interview where he spoke about it. I reached out to him multiple times. If anyone knows how to get a hold of him, let me know because I'd love to.
So he's talking about detoxifying glyphosate by taking glycine supplements.
Yes,
Oh,
yes,
yeah. Okay.
yes.
Well, I
Maybe
know
I'll try.
you're really you're a pit bull when it comes to tracking down the people who have some knowledge about things you're interested in.
Well, thank you. Thanks.
Yes, that is a compliment.
Thanks, Dr. Brind. And one other thing I wanted to mention before we go to, we have bunch of other myths to bust, but I'm ready for another Dr. Brind hypothesis because it was something I was reading. I know there's the glycine 1 receptor, and there's a few drugs that they're making that are like a glycine 1 receptor agonist, which then will increase the amount of glycine in the central nervous system, and it's related to these MMDA receptors.
Oh,
And I was wondering, some of them work. There's like, I think obidopertin or something like that. But I was wondering,
bioterin,
okay, there you go.
bioterin,
I was,
PTERI.
yes, BITOPRTIN.
The
Exactly.
P is silent, like it's subtle, right? The P is silent, like the B is silent, it's subtle.
Well, thank you. But I was wondering, so there's a specificity to that, right? That, they're increasing the glycine at a very specific place, potentially. But do you think, given the incredible safety of glycine, high doses of glycine, could you supplement high doses of
Yeah, but you
glycine?
can't patent that. See, that's why. That's the
Right, I
other
know.
problem with modern science is that it's all about drugs. So they say, "Wow, glycine is great for this condition and that condition." You go, "What a great drug!" No, I took, of course, the opposite point of view is to say, well, if it's so important for this condition, that condition, and the other condition, maybe the lack of it is what's producing the disease. So we're really talking about deficiency diseases, and I believe we are. But that's the problem is that it's all about, yes, we have to develop a drug because that's how you can get money to fund your research. So interesting, we were talking about sleep. So there are several good papers that came out about 10 years ago on sleep and specifics of, and this was Stanford University and a group in Japan working in collaboration and, doing these, electrophysiological studies in rats and mice by injecting it right into different areas of the brain. And you're thinking, gee, that's pretty sophisticated, expensive research. So who's going to fund research on glycine? People who make glycine. Ajinomoto is a Japanese company that makes amino acids. They sell glycine. Well, it's the Ajinomoto research group that funded that and actually did that research. So Ajinomoto, to its credit, does a lot of good research on the product it sells. But if you're not going to be in the situation where you're making money on the product, you know,
It's hard to do it. And that's why,
nobody's going to fund it. Now, I can make enough money to support the product and myself and actually make some more so I can advertise more and get the word out. But, I'm not going to make big money, from selling glycine, which you can't patent. So pharmaceutical companies are not going to be interested in that. But Ajinomoto, which is basically an amino acid nutritional company, they make glycine for pharmaceuticals too. That's the Japanese glycine, which By the way, Japanese glycine is made in America. It's actually made in Chattanooga, Tennessee.
Is it really?
Yeah.
Oh, that's fascinating. Well, and that was my thinking, not a physician, never medical advice, but I'm thinking if they're pouring this research into these glycine agonists, right, to increase the amount of glycine in that space, maybe we could take glycine and then not only get the
Maybe just, yeah, the supplement will do it not as efficiently. Oh, yeah, well, you'd need to take, maybe half a pound of glycine. Well, even if you needed to take half a pound a day, you probably could.
Yeah, or maybe we could just add in, 3 bone broths a day. You know what I mean? We could be
Couple of
eating more
ounces. Yeah,
glycine.
that's right. You could have, because when we say 8 grams, that's why I formulated sweetamine as a powder, because 8 grams is a lot of pills to take. That's a lot, but it's still only about maybe 2 teaspoons, a little less than 2 teaspoons for a bulk. That's still a bulk amount, but I am not advocating megadosing that way. I'm just saying this is what you need to get glycine up to where it should be if it were a normal, healthy level. And that's the biggest myth of all. You told me this was going to be about myth busting, but we haven't gotten to it yet. The biggest myth of all is that inflammation is a natural response to injury.
Yes,
and everybody still thinks that. Even glycine enthusiasts probably, because this is so endemic, it's so ingrained in our thinking and in medical thinking and going back for so long, even before our diet was as screwed up as it is these days. People were still glycine deficient. It's just gotten worse. It's gotten a lot worse because of dietary habits, because we eat the meat and we throw away the bones. We eat more and more and more meat and everything else, so we get fat. And when you get fat, your body makes less glycine. So, when you eat more muscle meat, you're wasting glycine that your body has. And so you're robbing your body of glycine, and you're throwing away the glycine you should eat. You're robbing the body of the glycine that it does have, and you're not making as much glycine as you should, because, so this is the epidemic of obesity is related to all manner of chronic disease in large measure because it exacerbates the glycine deficiency that is usually there in most of us. Not everybody. I mean, I had a chat with a colleague a few months ago. I'm always talking about glycine, right? Hard to shut me up, right? So I'm talking about the glycine, and she didn't know anything about it. The conversation got sort of naturally around to that. And I was talking about inflammation and so on and so forth. But before I got to that, relating it specifically to glycine, I said, I said, she had like a nice tan, a light kind of tan. I guess it was in the summertime. And I said, do you ever have any problem with sunburn? Because she was a fair-skinned woman, and she said, no, I don't have any problem with the sunburn. I go out in the sun all the time. I don't. And it turns out that she's a bone broth enthusiast. What do you know?
Yay.
She doesn't need sweetamine. She doesn't need a glycine supplement because she has something like a quart a day of bone broth. So you can get it from regular food, but the easy way to do it is you just take glycine, because we know from science, Occam's razor, which tells us the simplest hypothesis is probably the right one, and you reject hypotheses that are too complicated, not the ones that are too simple, then know that it's the glycine that's actually giving you the benefits, the key benefit of bone broth in terms of inflammation. And in terms of sunburn, of course, it's the inflammation following the sunburn that really does the damage. And because you think, your sun turns red. Oh my God, my skin's all red. Now you get out of the sun. Well, why is it not getting better? It just turned red. It didn't shrivel up and die and peel off right away. It didn't, right? It just turned red. So why can't that red color just go away? Well, if you have enough glycine in you, guess what? It just goes away.
You know what? And you're right, because I drink bone broth all day, but I just was in Arizona, 110, out in the sun. And I remember I did get a burn, but it didn't linger and it did go away. So that was actually this weekend. So
right. Well, that's because you're naturally so dark-skinned, actually. That's
exactly. Yeah. No, I'm Scandinavian. I should be.
I know. You're about as I would say you're more fair-skinned than I am, because at least I have freckles,
That's true. Yeah, I'm very, well, okay, that's beautiful. Yes, that is one of the biggest myths we have to bust, that you have to be inflamed just because you're aging, that's natural. I love that. Now, the other one that you always hear, that I always get asked about, is that glycine will feed, or that rapidly dividing cancer cells have increased reliance on glycine To what do you say about this, Dr.
Well,
Brind?
there's a lot of things to say. The research that showed that is very interesting for what it actually does show, because the research studies have shown that we knew about methionine, for example, many years ago, that cancer cells are more dependent upon methionine. So there is a treatment modality, Dr. Bob Hoffman, whom I know from many years ago, in California. I think he's in his 80s. He still has a clinic where he treats cancer with an enzyme treatment called methionine-ase, which actually gets rid of methionine so that you have very, very little methionine circulating in your plasma and in your body fluids, and so it's cancer cells their growth is suppressed because they are more methionine dependent than normal cells. Because remember, I talked about this methionine cycle. The body has a lot of metabolic machinery to recycle and regenerate methionine. In cancer cells, what happens is cancer cells are said to de-differentiate. They become more primitive. And the more primitive they become and the more advanced physiological functions they don't have to do, they don't have to spend the metabolic energy to recycle methionine, then they are more competitive as cancers to be successful in taking over the body, right? So why should they be methionine dependent? Well, because they arise and cancer cells are selected when cancer develops in a body over a period of years. The cells are selected like in a Darwinian process. The ones that are fittest will survive better. Well, what's most fit in a body that is overly enriched in methionine? Well, it's going to select for cancer cells will grow better because they do not waste the metabolic energy to recycle methionine. There's always plenty of it around. So it's not surprising that cancer cells should arise, not all the time, but probably a majority of human cancers tend to be more methionine dependent because they arise in a body that is methionine rich. In the opposite direction, what about glycine? Well, it turns out that cancer cells, most of this work was done with breast cancer cells, are better at utilizing glycine because they arise in a body that is glycine poor. So if your glycine levels are low, what cell is going to outcompete normal cells and take over the body as a cancer? One that can get by with a lot less glycine. So cancer cells can use glycine more efficiently. But what does that mean? Oh, if you take glycine supplements, then isn't that going to make the cancer cells grow more? And I say no, because the fact that they can use glycine more efficiently means that they will use lower levels better than normal cells will. So you pretty much have to keep it down to such a level that you kill all your normal cells before you hurt the cancer cells. Cancer cells will be more resilient to low glycine levels. They can grow better because cancer cells develop naturally or in a body that's out of whack metabolically because of diet and lifestyle factors, such that they grow well, they arise in a body that is glycine poor and methionine rich. So what do you know? That's what they're finding out about these cells, is that they grow better in an environment that is glycine poor and methionine rich. So you try to make the environment methionine poor, but glycine rich is not going to have any effect because your normal cells will do better with more glycine. Now you can say, well, didn't they say take cells and grow them in an environment that was low in glycine or glycine free actually, and give them glycine and the cancer cells grew more rapidly? Yeah, but if you look at the small print, you actually look at again the levels, the concentration. How much are we talking about? The glycine that they gave that made cancer cells grow better was 100 micromolar. That's actually below the lower limit of normal of glycine, which is about 150 micromolar. So yeah, you give them very little bit of glycine, they will grow like crazy, but the normal cell will not grow so well in 100 micromolar glycine. It needs more. So that's why that sort of doesn't add up if you kind of do the theoretical arithmetic. In real world, what glycine does, we do know as far as the immune system related to cancer, and also, well, it's not really the immune system, but the one effect that we do know of is that glycine receptors are also present in endothelial cells. That's the lining of the arteries, which is also the layer of cells that forms new blood vessels, new capillaries. So when cancer cells develop, one of the things they need to grow into like a substantial solid tumors, they need to grow new blood vessels. And what glycine does is it suppresses the growth of new blood vessels. It regulates that, sort of like it regulates platelet aggregation. So that glycine is anti-angiogenic, is the term that's used. So, for example, this was shown in a paper from, I don't know, probably around 2000 or so, 2010 maybe, that showed that if you take a standard transplantable tumor, the B16 melanoma is a mouse melanoma that is used to study cancer in mice, and you can passage it from one generation to the next by taking some cells and injecting them into successive generations of mice. But if you supplement the mice with glycine, guess what? The tumor cells won't grow. That was actually corroborated by a colleague of mine. He didn't publish it, but he corroborated it. And then we corroborated it in our study with longevity in mice. When you supplement them with glycine, yes, the mice all live longer. And why did they live longer? Well, the main cause of death was actually lung cancer. The cancer... It's not that glycine is going to prevent cancer or anything like that, because cancer is one of the ways that old people used to die of old age. The horror of modern cancer is that it happens to younger and younger people. Someone was, 89, 98, whatever, age, and so on, they died of cancer, and that was typical. It was, it was a disease of old age. When I was a young scientist, cancer was always a disease of old age, viewed that way. These mice, in our studies, they mostly died of lung cancer, lung tumors. It was just the way that the mice died when they were, too old. And the specific reason why they lived longer was because the lung tumors, which eventually they succumbed to, grew more slowly. Because what would happen is they would get tumors in their lungs, and the cancer would grow and grow until eventually they couldn't breathe anymore and they would die. And the tumors took longer to grow. Again, presumably because of this anti-angiogenic effect of glycine. So in the real world of cancer and human cancers, most likely, it's kind of theoretical putting things together, most likely glycine would slow the rate of cancer growth. Not like it's a good chemotherapeutic drug, that you could rely on, and it is not specifically going to target cancer or stop cancer from growing, but it has this anti-angiogenic effect, so it probably slows the rate of growth of tumors. But the idea that it will cause cancer is, these were a couple of papers that came out in 2009, and especially there was a paper that came out in, I think it was maybe 2011, that looked at prostate cancer, and it said, glycine makes prostate cancer grow. It makes these normal prostate cells turn into cancer cells. First of all, the cells that they were using were not normal cells. Because cancer cells are immortal, in a sense. You can take normal cells in culture and passage them from one generation to the next, to the next, to the next in test tubes. But eventually they will die out after 20, 30 generations, whatever. Something called the Hayflick number. I'm not going to get into that, but they will die out. They are mortal. One of the things that cancer cells are is they are immortalized. In other words, you can go to the American Type Culture Collection and you can still buy HeLa cells or various cells of prostate cancer. This was about prostate cancer, that have been around for decades and decades, and keep passaging them, which is great for research. It's very good science because it's the same cell line. But the thing is, they are used, this particular cell line, it was called RWPE cells. They're used in tissue culture as a stand-in for normal prostate cells. Guess what, though? They're not normal. They're not normal, and yes, they are converted into cancer cells with any of a number of different kinds of stimulation. One of them being excess androgens, like testosterone, of course, that will make them grow, or also something called sarcosine.
Yeah.
And sarcosine is a metabolite of glycine. Remember I talked about glycine N-methyltransferase to get rid of excess methionine. Well, the immediate product of glycine N-methyltransferase is sarcosine. Sarcosine doesn't last very long. It gets gotten rid of right away. And when you put in a lot of sarcosine into these cells, guess what? They become malignant, or they act in a malignant fashion. The paper also said that glycine, that glycine will do. Glycine and sarcosine, when you give it to these cells, they get really invasive and malignant. So, of course, that was the headline, right? That was the University of Michigan. It wasn't Cleveland Clinic, but it was a big reputation medical center, research center, and it was picked up by popular media And so that was the whole, glycine makes prostate cancer grow. But then it was about 5 years later, another paper came out, and they said, right at the bottom of the abstract where they give their conclusions, they said, and we found that in this system that sarcosine, but not glycine, induced the malignant phenotype.
Mm-hmm.
3 key words, but not glycine. It was kind of like a retraction, but very key. This is important. When I took 40 grams of glycine a day, by the way, to see if it would do me any harm, and it didn't. That was how long ago was that? Almost 20 years ago. My sarcosine, the most my sarcosine ever came up to was 2 micromolar, but they needed something like 20 micromolar sarcosine to show any effect. So again, it's big amounts of that, big amounts of androgens, big amounts of certain things that would have this particular effect on this particular experimental system. So that's a problem with experimental systems, science. Is the science good science is one question, but even if it is good science, what's its relation to the human situation in health and disease? That's what's called translational research. We found this in cultured cells, we found it in a test tube. What does that mean in terms of patients who have this disease or who might have this disease? And that's a whole different question, and you can't just jump from one to the other. As far as the cancer claims go in terms of breast cancer and prostate cancer and glycine, and I brought in the findings about methionine because, again, it's that glycine-methionine connection showing up in the nature of cancer cells that develop in our modern society. What kind of cancers do you get? You get cancers that are more methionine dependent and less glycine dependent because they arise in a body that is glycine poor and methionine rich. So that is exactly the kind of cell that's going to be selected for as a successful cancer.
Yeah. And there's also, an inflammatory component and a metabolic component, at least to some theories, that cancer might thrive in, metabolically dysfunctional environments and inflammatory environments, and glycine could potentially help both.
Oh, yes, of course, yes, that's true too. Yeah, the origin of cancer, that's right. Cancer will, of course, thrive in an environment that is inflammatory. Many cancers, so this goes back to some of the older research that I've mentioned in other shows that we did, that you have, for example, cervical cancer. It's been known for 40 or 50 years that women who have more ovulations in their lifetime have a higher incidence of cervical cancer. Not cervical cancer. Ovarian cancer, sorry. Because the act of ovulation is actually something that does violence to the ovary. The ovary shoots an egg out of a cannon, and that does damage to the surface of the ovary. Some women feel it every month. They call it Mittelschmerz, that pain. And that's because it's a very violent thing that happens in the ovary, and it's once every other month for each ovary, so no big deal. It's an injury, but it heals. But if you're glycine deficient, you get inflammation associated with it. And so that results in more women who have, the more ovulations you have, the more likely you are to be one of those women who ends up with ovarian cancer from it. So yes, the inception of cancer is definitely going to be reduced by having a lot of glycine around, because cancer is one of those things that happens from inflammation, and that's been shown. Even, for example, I was mentioned prostate cancer. There's even some research that shows that a lot of times prostate cancer will be suppressed by androgen deprivation, and then it will come back years later. All of a sudden, now you have a cancer that's been selected for not needing androgens in order to grow and multiply and invade other tissues. And it seems that one of the things that drives this epigenetic conversion of the cells into a more aggressive, less dependent phenotype, so that it's more dangerous and more lethal, is inflammation. It's part of the inflammatory cascade. These things are needed. That's what drives the cells, the cancer cells, to become independent of all these other control mechanisms. Because then you have a cancer and the cancer is under control, but you have the same diet and the same condition of the body biochemically. Eventually, you're going to select for something that expresses a malignant phenotype again down the line. So in so many ways, it's really clear that Glycine is going to be an anti-cancer rather than a pro-cancer or a pro-carcinogenic influence in the body. And it's natural. You need glycine. You need a lot of it. Your body makes a lot of it. So let's not think too negatively about things the body actually needs. In terms of cancer, like you can always cure cancer very easily by starving yourself to death, right? So then you and the cancer both die, right? So.
Perfect. I think I love that. That is a perfect way to wrap that section up. And there's only one more myth I want you to bust.
Okay.
And it's about MTHFR and methylation, your specialty.
Yes.
So
Yeah. That is.
MTHFR should not be supplementing with glycine, Dr. Brind, and that it won't help them.
MTHFR should not be
Supplementing with glycine, that it won't.
Who said that you shouldn't be? Why should? Oh, yes. Now I'll go back to my original experiment that I published and I talked about in 2011 on mice and longevity and the reciprocal relationship between methionine and glycine. So because of that, you would think that methionine is the main methylator. It's activated, it becomes SAMe, S-adenosine methionine. That's sort of cocking the trigger, cocking the gun so that the bullet, which is S-adenosine methionine, can actually shoot that methyl group onto another compound. Okay, so we thought that in order to mimic methionine restriction, because it was beneficial to longevity to restrict the amount of methionine in the diet, that we could simulate that by adding glycine to the diet, which would help to strip methionine. And what we found was, yes, it did increase the lifespan of these animals, but no, it had no effect whatsoever on the amount of methionine in their blood. Isn't that interesting? Mm-hmm. So it did not accelerate the clearance of methionine. So what are you supposed to do? So if you have an MTHFR mutation, like the common one, which is C677T, if you have it, you know what I'm talking about. If you don't, it probably won't interest you. But so if you have that mutation, one of several mutations, but that's the most common one, it is a mutation in an enzyme called MTHFR, which stands for methylene tetrahydrofolate reductase. What that means is that methylene tetrahydrofolate reductase needs to be converted to methyl tetrahydrofolate reductase. Methyl is that key word, right? Because that methyl group gets added back to homocysteine, which is what happens to methionine after it's lost its methyl group. It's now called homocysteine. You stick that methyl group back onto homocysteine, now you have your methionine regenerated. So that's the key in the methionine cycle. The key enzyme is to make folic acid methylfolate, and that is capable of remethylating homocysteine, which regenerates methionine. So if you take glycine, remember we found that taking glycine does not help you to get rid of methionine. Oh well, we hope that it would, but it doesn't. So eating glycine is not going to affect your methylation status. So what are you supposed to do then if you have this problem? And also, from a scientific point of view, how do you explain it? The whole dynamic that's going on. So let's explain it. You have a methionine-rich meal, a cheeseburger. I don't know why I always think of that. That's
You do.
my favorite methionine-rich meal, I guess.
Chicken breast.
So you have that cheeseburger, and and in the next hour or two, as I said, in the next hour you're digesting and absorbing it, and an hour after that you're busy, incorporating it and basically getting rid of the excess methionine. If you have an MTHFR deficiency, that's all going to be normal because you've taken in enough methionine now for your body to have and be able to recycle it over that period between one meal and the next, right? Well, if you have this deficient enzyme, that means you're not going to be able to recycle the methionine. So, now your body's gotten rid of it, now it's got to recycle it 2 hours after you ate. Now I'm low on methionine and I feel faint, I have no energy, I have all of these things that low methylators have, and they all know that they have to have some kind of a high protein snack in between meals. I have a family member who's like that also. There's somewhere in my family there is that mutation, it seems to be. And there's lots of things you can do about it. Well, first of all, what's deficient? Methylfolate. You can take methylfolate. That's number one you can take. Number two, you can also get around the inability to recycle methionine through the methionine cycle by going through the alternative cycle, which is called betaine homocysteine methyltransferase, which is betaine. What's betaine? Commonly known as TMG. So you can take trimethylglycine, or you can just take more methionine in between meals. What's the best source of methionine for a snack? I say Brazil nuts. 3 Brazil nuts is 100 milligrams of methionine. So you can take Brazil nuts for a snack. There's your high protein snack, give you more methionine. Or you can take TMG, or you can take betaine. I'm sorry, betaine is TMG, or you can take methylfolate, and you can try any of these, all of these, some combination of these, whatever, if you have that mutation. But again, I'm talking to you. If I'm talking to you who have this mutation, you know that there's that period where your energy really goes to pot. In that 3-4 hours during that fasting period between, let's say, lunch and dinner, between 12 and 5, right? If you have it at between 2 and 3, you have a high protein snack. Well, if that snack is, I would say, have 3 Brazil nuts, or have your TMG supplement then, or have your methylfolate supplement then. So time it right. You're always going to have lots of methionine right after a methionine-rich meal. Like everybody else, your absorption of methionine is not abnormal. Your processing of methionine and using it to methylate things is not abnormal. What's abnormal is the recycling of methionine. So you can fix that. As I said, the 3 easy fixes, easy over-the-counter fixes. My favorite.
I love that.
So
That's great. So to sum it up, because people have been very confused on this topic, taking glycine is not going to hurt methylation.
No.
Okay, it potentially, if you're taking TMJ, this is actually another one of the questions. Would that be considered TMG? Would that be considered an adequate source of glycine?
No, because you're taking it for its methyl groups, which is much less. You're not going to take 8 grams of TMG.
Okay.
Some 100 milligrams, maybe, or something like that. Oh, and one other corollary I should add, because they say, oh well, to get more methionine, let me take the most active component. Let me take SAMe. SAMe is not only more expensive, but SAMe, S-adenosyl methionine, is mostly adenosine, which is a lot heavier than the methyl group, which means that most of SAMe is adenosine. Most of it is not methionine. So to get the methyl groups, you would need less of if you need 100 milligrams of methionine, probably 50 milligrams of TMG, and less than that, maybe 5 milligrams of methylfolate would do pretty much the same thing if you do the arithmetic on how many methyl groups are going in and out. But basically, these are things that are pretty much harmless over the counter. But SAMe is not necessarily harmless. SAMe is very reactive. But if you take SAMe, it's very expensive, very inefficient, because you're mostly not taking methionine, you're taking the adenosyl group, and it's potentially can cause problems. Mental problems also. It's been associated with bipolar disorder. It can really generate a manic episode, because SAMe is a very potent biochemical. So I would say avoid SAMe, but just plain methionine, largely in the form of Brazil nuts, is probably the best. Trimethylglycine, otherwise known as betaine, or methylfolate. So many ways to get around this mutation. And the reason why so many people in the population have this mutation, and I'm saying it's somewhere between 10 and 20% of the population, depending on your ethnicity, is because it's not really that serious a mutation. It's more of a nuisance, Not a really, say, is it a disability that you really don't have the energy to work optimally between lunch and dinner, and you need a snack in between? It's really more of an annoyance, but it's a very easy fix.